Okay.
Okay, can I go?
Yeah, you can go.
Yeah.
Thank you, Anna.
Great, thank you. So good afternoon, everybody. My name is Anna Zampetaki, and I'm a lecturer in the Cardiovascular Division. I would like to welcome you all to this BHF Center Technology Workshop on CRISPR-Cas9 Gene Editing and Cardiovascular Diseases. What we're going to talk about today are the basic principles of gene editing, how to design a CRISPR-Cas9 experiment, CRISPR-Cas9 applications beyond gene editing, and therapeutic approaches using the CRISPR-Cas9 system. Now, the clustered regulated interspaced short palindromic repeats, or CRISPR in short, created such, quite a stir in biomedical research. Only in 2015, the journal Science declared that it is the breakthrough of the year. And stated that CRISPR is posed to revolutionize research. And just a few weeks ago, the two scientists that developed the precise genome editing technology actually got a Nobel Prize for it. However, CRISPR actually comes from very humble beginnings. It was invented by bacteria to combat viruses. So what is happening when a virus is infecting a bacterium? You see the foreign DNA here. So the bacterium will cut the DNA in pieces and insert it into a specific locus, the CRISPR locus. Now, this locus will then be transcribed and give a precursor CRISPR RNA. These RNA molecules are then cut into smaller pieces and loaded into protein complexes that can degrade the foreign DNA. So the RNA and the protein complexes will bind to the foreign viral DNA and degrade it. There are several types of CRISPR-Cas system, but by far the most popular and best studied is the type 2 that encodes the Cas9 nuclease. And the reason is very simple, because the requirements for this system are minimal. So what you have is the Cas9 nucleus. It is shown here in blue. It actually has two nucleus domains. It's two scissors. These two scissors, each of them will cut one strand of the DNA. And it will generate a double-stranded break. To cut, it is guided by a small RNA molecule, the CRISPR RNA, shown here in yellow. This is only 20 nucleotides long, and it is complementary to the site that is targeted. Apart from these 20 nucleotides, you need adjusting a PAM sequence or a motif for your Cas9 from Streptococcus series NHEJ. And this is really all the requirements of the system, apart from an adapter molecule, a tracker RNA that is needed to create a complex with your CRISPR RNA. So you see that the requirements are very, very low. What was very convenient was that scientists could actually generate the fusion of CRISPR RNA and tracker RNA. And by doing so, they generated what we now call the single guide RNA. In this single guide RNA shown here, it's very easy to produce synthetically. All the companies can do it for you. Also, the nucleus Cas9 again would be available, and therefore it is a system that is easy to use. Just to make sure that everyone is on board, for the CRISPR-Cas9 system, we need the Cas9. In most cases, Cas9 from Streptococcus pyogenes is used. That is about 160 kilodalton, and this has to be introduced. We need a single guide RNA molecule. This is about 120 nucleotides long. Again, this is exogenous. It has to be introduced in the system. And we need the PAM sequence for Streptococcus pyogenes is NHEJ, and this is present on the DNA. It's this red part that you can see here. So how does this compare to the previous methods we had for gene editing? Well, the previous methods that we had were protein directed. So you have zinc finger nucleases. You have the zinc fingers that identify nucleotide triplets. So a sequence of zinc fingers, and then the FOC1 cleavage domain at the carboxy terminal, both right and left zinc fingers, and a double-stranded break. For your talons, you actually have 4 talons, one for each base pair, and again you have the cleavage domain, the FOC1 at the C-terminal. So both methods actually require protein engineering and are more advanced in terms of molecular biology required to generate this specific protein targeting your gene of interest. If you actually compare the different platforms, you will see that the efficiency and the ability to multiplex for CRISPR is much higher compared to the other two. But additionally, the chance of off targets is higher on CRISPR compared to the other two. And the reason is quite simple. You have 20 nucleotides that actually identify the targeted region. It is likely that there is some homology in these 20 nucleotides with other regions in the genome. But we'll discuss a bit later about the off targets. What I want to make clear is that CRISPR is an RNA-directed gene editing, while zinc finger and talons are actually protein-directed gene editing platforms. So what is happening once you have the double-stranded break in your cells? Well, the cell can actually not afford to have the DNA cut in half. So it will very rapidly repair the double-stranded break, and in doing so, it will make errors. You can have deletions as shown here in red, or insertions here in green, but this is the default. The double-stranded break will be very rapidly, the two ends will be stitched back together. If you actually have available a donor template that has homology in both the left arm and the right arm with the region where the double-stranded break occurs, as you can see here, then you can have also the homologous-directed repair. So in this case, you can have precise insertion and modification on the side of double-stranded break. I have to say, however, that the homology-directed repair is much more inefficient. Usually 1 or 2% of the cells would only be repaired with the homology-directed repair, even in the presence of the donor template. All right, so let's move now to designing CRISPR-Cas9 experiments. And the single most important question that you have to answer is, what is it that you want to accomplish? CRISPR-Cas9 is a tool that can be used for various things. You can use it for knockouts or knock-ins, to edit the gene, knock down a gene, activate, or for very different reasons like imaging, for example. So it is important to know what is it that you want to do initially. You can use it for gene knockout, so you can use it as a hammer. When homologous end joining is what the cell will do for you. In that case, you need your Cas9, your nuclease, the scissors that will cut the DNA, and also your guide RNA that will direct the complex to your target of interest. Here your PAM is shown in purple. You have the double-stranded break, and then you let the cell repair the double-stranded break. In doing so, it may generate frame shifts, premature stops, splice disruption, or regulatory element deletion. This will heavily rely on the cellular mechanisms of DNA repair. Or you may want to try something that is more elegant and do a knocking or editing while introducing specific elements. In that case, you will have to do homology-directed repair. Again, you need your Cas9, your single guide RNA to direct your Cas9 and RNA complex to the target of interest. Once you have your double-stranded break, however, you will have to provide your template that will be used for homology-directed repair. In this case, you will introduce a very specific sequence. But of course, there are additional requirements. It would be good to also introduce a mutation in your PAM sequence so that your insert is not cut again by your Cas9, and also the distance between the single guide RNA recognition site and the mutation is critical for the efficiency. Now, you may also choose to do gene inhibition or gene activation using CRISPR-I and CRISPR interference and CRISPR activation respectively. We said that our Cas9 has two scissors or two nucleus domains. These domains can actually be mutated, and then your Cas9 will have no nucleus activity. In this case, we say that we have a dead Cas9. And this dead Cas9 can be fused with a repressor domain, for example, CRAB. And by doing so, it will still bind to its single guide RNA and be directed to the DNA at the site of interest. But by doing so, it won't be able to generate the double-stranded break. On the contrary, what you will have, your CRAB will induce a chromosomal modification leading to gene repression. In a similar manner, your dead Cas9 can bind to your single guide RNA if it is fused to activation domains like VP64, for example, then it will lead to activation of the gene of interest. All right, so having all that in mind, let's say we have decided what is it that we want to do, and now this is the checklist for our gene editing experiment. So we have lots to do. First of all, we have to design the guide RNA. There are software programs to do that. We have to then decide what biological system to use. will it be in cells or in animal models, what delivery method to use, and what would be the mutagenesis detection method that we will employ. Now, when it comes to the single guide RNA, we said that our guide is 20 nucleotides long. Actually, within the guide, there is this sequence that is close to your PAM sequence, and this has the highest specificity. Normally, your Cas9 will cut 3 or 4 nucleotides from the PAM sequence, and this is the area that has to have almost absolute homology for your Cas9 to cut. Otherwise, the cut won't happen. However, in positions 1 to 7 or the lower specificity, you actually can have mismatches that are tolerated, and your Cas9 can cut the DNA despite the presence of mismatches. Therefore, as a safeguard, we would always choose single guide RNAs that are off targets with no PAM sequence. If there is no PAM sequence, your Cas9 cannot cut at all. So definitely no PAM sequence for all your off targets is highly desirable. If there are other sites or off targets with some similarity to your target of interest, then definitely try to have at least 3 mismatches in the length of your guide, and also if possible have 2 mismatches in the proximal region right here, because if the mismatches occur here, your off target will not be affected. Now, there are several programs that you can use to design your guide RNAs. I would recommend that you use the CRISPR Finder from the Sanger Institute. And this is how the homepage would look. You can actually copy and paste the sequence that you want to target. And let's say, for example, that we want to target the human HDAC3 gene. So in here in Ensemble, you see how the genomic sequence looks. In red, you have the exons of the gene, and in white, you have your introns. And here I have highlighted in yellow the ATG, the translation initiation site. So what I suggest that you do is you actually copy sequences upstream and downstream of the ATG and use that as an input sequence for your CRISPR finder. This is what we've done here. So this is your sequence. This is how the results would look like. You have a complete list of the single guide of the guide RNAs that you can use to target this region. Now, if you actually click on the first one, you will see the sequence of the guide RNA, the PAM sequence, and the orientation, whether it is on plus or minus strand. And here you will also get a list of the potential of targets with the mismatches highlighted in red. Now, as you can see here, there are several off-targets, potential off-targets that have 2 mismatches. So this is not ideal. All guides are not created equal. I would strongly recommend that you only choose guides that have at least 3 mismatches in any of the potential off-targets. Therefore, I would have gone back to the list and actually check out a different guide RNA, the one that I have highlighted here. If you have a look here, you only have 1 potential off target with 3 mismatches, and you see one of them is actually in the proximal region, well, both of them, these 2 are in the proximal region, so there's no way that they will be targeted by my guide. So this is a way to actually make sure that your gene editing is as specific as possible. Now, things are a bit different when you want to design single guide RNAs for non-coding RNAs. And just to remind everyone, we all know from school that DNA is transcribed into RNA, and this is translated into protein. But following the ENCODE project, we know that now DNA can actually transcribe into RNA, and this RNA may not encode for a protein, but may still have regulatory function. So you can have longer coding RNAs, more than 200 nucleotides long, or small RNAs. These I'm showing them precursor for a microRNA, a hairpin RNA. In that case, for example, here for microRNA 195, I would recommend that you actually go back to your ensample and find out the sequence of your precursor microRNA. Here it is highlighted in red. And in this case, I would recommend that you actually design guides that target specifically the transcript. You see here the PAM sequence is shown with blue. This is where your PAM sequence lies, and the arrow shows where the double-stranded break will occur. A different guide, again, the PAM sequence in blue. This is where your arrow will cut your transcript. If you want to work with long non-coding RNAs, because these are longer, what I would recommend that you do is use two guide RNA. So you have a dual system, generate one DNA cut upstream, one downstream of the transcription initiation site, and create a truncation that will lead to no transcription from the specific site. This is what I'm showing you here. So just by doing a genomic PCR in the locus, you can right on the spot identify whether your editing works. As you can see here, we have a truncation. Here we have different guides. The truncation length, a truncated product is different as well. And what is nice about it is that if you do Sanger sequencing in these fragments here, you will see that the cell tends to stitch back the ends together. So you have, in most cases, a perfect religation and well-defined sequence for the spot. All right, so we designed our guide RNA. It's time to decide which biological system will we use. Will it be cells? Will it be an animal model? This is important because it will actually define the delivery method that we will have to do. For the delivery, we have a lot of choice. You can use plasmid vectors that are inexpensive, but then again, not all cell types, for example, will give you high transfection efficiency. You can use lentiviruses. They can be used for multiplexing, library preparation. They're not very expensive. However, they do integrate in the genome, so there may be safety concerns. Or you can use mRNA for Cas9 and RNA for your synthetic single guide RNA. These are great for injections into the zygote, but they are quite expensive, and the stability of the RNA components may also be affected. What is also very popular is using recombinant Cas9 protein and a single guide RNA molecule, so that again you have a transient expression. Your Cas9 does a double-stranded break, and then within 24 hours it is cleared together with the RNA, so it's much safer. But again, it's very, very expensive, and there is an issue of the stability of the RNA components. Additionally, you can use adeno-associated viruses, small single-stranded DNA viruses. that have specific serotypes or specific organs can be targeted. They are great because they will have very low immune response, but unfortunately they come with very low packaging capacity, which effectively means that if you want to use AAVs, your best option is to have two viruses, one that will encode your Cas9 from Streptococcus pyogenes, the other virus encoding your single guide RNA, so that you also have regulatory elements in these viruses. Or the alternative is to use a Cas9 from a different species, from Streptococcus. But please note that in this case, your PAM sequence will also be different. All right, so you decided on the delivery method and went ahead to done the gene editing. How do you actually then quantify your mutagenesis? How do you detect mutagenesis? Well, a good idea is always to design guides that, if possible, disrupt a restriction site. Here in yellow, you can see that this site corresponds to SSP1 restriction enzyme. And when you have edited cells, what you can get is different patterns in the restriction digestion products. As you can see here, all these are edited samples, and the pattern is different from the unedited. This is the first readout, but of course it's not quantitative. You can quantify your gene editing using a T7 endonuclease 1 assay. So in your edited cells, you will always have cells that are wild type, unedited, and mutated cells. As you can see here, the mutation is highlighted in red. If you actually do a PCR for your area of interest, let's say that these are the PCR amplicons, and then denature them, you'll have single strands. And these single strands can be either wild type or mutants. If you then re-anneal them slowly, you'll have random formation of dimers. And you can have a wild type dimer, or a mutant dimer, or a heterodimer mutant and wild type. Now, this heterodimer will actually form a mismatch. And these mismatches is what your T7 endonuclease 1 will identify and actually digest. So you will have a cut in these mismatches, and this will look like this. So in the same cells that we actually did the restriction digestion, we use an amplicon for T7 endonuclease 1 assay, and you can see here these are edited cells, and these are the digestion products from the T7 endonuclease. There is no digestion, of course, in the unedited sample. You can quantify these bands, and based on the intensity of the uncut band, we found that you have almost 30% editing efficiency, which is actually quite good. It is important that you also assess the off targets of your guides. You never know whether an off target is indeed happening, and therefore I would recommend that at least the top 10, 5 or potential off targets are screened and also assessed by T7 endonuclease 1, as we did here. Now, we relied on non-homologous end joining in our experiment, and you can actually have a look at the sequences that occur in your cell population by doing a TA cloning, as you can see here. So these are single colonies. If you pick these single colonies and sequence them, then you will get something like that looks like this. You have several deletions and in our case only one insertion, but you can appreciate that you actually have a vastly different population in there that leads to gene down regulation as you intended, but be aware that it is not a pure population. If you, on the contrary, want to go for something that is more precise genome editing is possible by using an homology-directed repair template, as you can see here, so that you can do gene correction, SNP modeling, conditional alleles, epitope tagging, and fine mapping of regulatory elements as well. This HDR template, of course, has to have some specifications. And ideally, what you want to change, delete, or insert will be small, like 50 base pairs changes or point mutations. In this case, you can actually use a single-stranded DNA oligo as a donor template. They typically come around 100 and 150. You have two arms, so you have an homology arm, left and right, and ideally you introduce the mutation somewhere in the middle and as close to your double-stranded break as possible. If you intend to actually make bigger mutations, deletions, or insertions, then you definitely need a plasmid vector that will have homology arms of 800 base pairs on each side. And I have to stress again that the HDR is really ineffective. So you will end up with a mixed population that you'll have to select and clonally expand. Now, the unfortunate thing about the HDR is also that it is restricted to the late S and G2 phases of the DNA, simply because that's when your sister chromatids are available to serve as repair templates. So people have actually tried to use drugs to cell synchronize their experiments so that their HDR is enhanced, and there is some effect, but not dramatic, I'm afraid. Also, you can optimize the single strand oligonucleotide that you use. So you see here you have asymmetry between the left and right homology arm, but in any case, it is good to keep in mind that the percentages are still low. Recently, just last year, a new method came across for precise editing, the prime editing. And in this case, what you have is a nickase. So a nickase is actually a Cas9 with only one nucleus available. So one of them, it can only make a nick cut a single strand on the DNA and not cut both. And this is fused to reverse transcriptase. And instead of having your single guide RNA, the 120 nucleotides long, you actually have a much longer RNA, the prime editing gRNA, that contains not only the binding site to the DNA, but also the RNA for the site that you want to insert. So this RNA will actually be used to generate the cDNA by your RT, your reverse transcriptase, and this cDNA is what will be inserted as new DNA in the locus. After original sequence excision, you will end up having a fully edited DNA. So the great thing about that is that in theory, it can correct 89% of unknown genetic variants that are associated with human disease. So that's fantastic news. But however, we cannot overlook the fact that there are extremely big challenges. This is a very sophisticated system. It is multi-step. Imperfect random edits can occur, and you can only make small indels, really 5 to 80 base pairs, and also it does require a very large prime editing system. But nevertheless, based on the current evidence, it is, compared to the HDR, more than 270 times effective. So that is perhaps an alternative. I would expect that the platform will be developed further before it actually reaches any use on a routine basis. Now, let's move on to CRISPR-Cas9 applications before genome editing. It is important to understand that your Cas9 without its nuclease activity, or your dead Cas9, is actually an RNA-guided DNA binding platform. It is an enzyme that can bind to the DNA at a very specific manner because it is guided by a specific defined RNA. And by binding to this specific loci, it gives us a lot of opportunities. You can actually have a fusion of your dead Cas9 with a number of proteins that are involved in histone modification, for example. So you can have histone acetyltransferase or deacetylase or methylase, and induce acetylation, methylation, ubiquitination, anything of your histones in a very specific manner. You can also have Cas9 that is fused with TET, for example, or DNA methyltransferases, and by doing so, controlling the DNA methylation status of your DNA. You can fuse your dead Cas9 with fluorophores, fluorescent proteins or tags, so that you can use it for imaging. And as we already mentioned, you can have activators and repressors, so that you activate or repress the gene of interest without doing gene editing. So the possibilities are actually endless, seemingly. The other thing that is important to mention is that your Cas9, when it is catalytically impaired, in this case they used an E-case, and it actually fused to a base editor. So a base editor would deaminate cytosine on uracil and would actually make point mutations from C to T or G to 8 is an additional tool that you can very specifically introduce mutations without the need of an homology-directed repair. And this can be important because the DNA-based editing can correct over 25% of the human pathogenic SNPs, and we will discuss a couple of examples where they actually used this technology. Now, moving on to therapeutic approaches using the CRISPR platform, the very first pathology disease that was targeted was the Duchenne muscular dystrophy. So muscular dystrophy occurs because you actually have mutations in the gene encoding this dystrophin. So this is a huge protein that is actually a link with the extracellular matrix and the cytoskeleton in the cell. And so far, really lots and lots of mutations have been identified. Others lead to truncation of your protein. or you may have an introduction of a stop codon. In any case, all these mutations lead to muscle degeneration, weakness, and myopathy. The Duchenne muscular dystrophy was a target of intense investigation. And really remarkable studies were published how even postnatally you can actually improve and partially restore dystrophin expression in the mice in vivo and also using different approaches. One thing that I would like to draw to your attention is this study here, where they actually kept the mice for longer and observed them 1 year on. And as you can see here, they found that AAV CRISPR is immunogenic when injected into adult mice. That was perhaps a bit disappointing, but what the authors propose is that if you do it in neonatal mice, you actually had a better outcome even 1 year later. Now, perhaps a good alternative is to use nanoparticles to deliver the Cas9 ribonucleoprotein complex. We said that you can actually deliver the recombinant Cas9 and RNA straight away. And in this case, what they used was these gold particles where they have the donor DNA Cas9 and the gRNA, the guide RNA. And as you can see, they actually corrected stop codon leading to restoration of dystrophin expression in the muscle. I just included here for you the SDR frequency, and you see we are talking about 2% if the dose is really high, 6%. So really low percentages, but nevertheless sufficient to make a functional impact on the muscle. Now, the other case that I would like to discuss is the type 1 diabetes mellitus study. So this study came out very recently. It involves, it focuses on the Wolfram syndrome. Now, these individuals actually have a mutation on this gene here, wolframin. This is it is believed to play a role in calcium, and when you actually have these pathogenic variants, then you end up with ear stress and unfolded protein response, and that effectively leads to beta cell death. So it is a diabetic condition. What they did was use iPS cells from these patients and then differentiate them, gene edit them to correct the mutations, and then differentiate them to beta cells. These beta cells were then injected into diabetic mice, and now because these beta cells actually function well, the cells were no longer diabetic. This is a good workflow to suggest that it could perhaps be done with, of course, with several modifications in humans as well. Now, another very exciting idea is this idea that you can have vaccination against heart diseases. PCSK9, I'm sure most of you are already aware of this protease that binds to the LDL particles and the LDL receptor. This is shown here in yellow. And it effectively directs the complex to the lysosome. By doing so, it doesn't allow the LDL receptor to recycle to the cell surface. And by doing so, you have less receptor available to pick up the LDL fragments, and therefore the LDL particles uptake drops, leading to high levels of LDL. So far, it has been targeting using antibodies, as you can see here. So by binding the antibodies, by binding to the PSK9, actually allow the LDL particles to bind to the receptor, and then the receptor to be recycled to the cell membrane. And very remarkably, these antibodies led to 50% reduction in LDL when used in combination with statins. 70% of LDL reduction was observed. So definitely very high interest in the cardiovascular field for this gene. What they have done is use this base editor that we talked previously to inject adenoviruses that target the PCSK9. And as you can see, this leads to reduced levels of expression, and the cholesterol in these mice, the cholesterol levels in the plasma are also reduced. Now, in a similar manner, angiopoietin like 3 protein, it functions in a similar manner by inhibiting lipoprotein lipase. That is involved in triglyceride metabolism. We know that actually mutations that occur normally have no adverse health consequences, and inhibition of this gene would be ideal with inactivating mutations. So initially what they have done in 2 clinical trials is use antibodies against angiopoietin like 3, and you see a vast reduction in triglycerides in healthy individuals with mild or moderate levels of triglyceride and LDL cholesterol. While in a group of homozygous familial hypercholesterolemia, again, the LDL cholesterol levels dramatically dropped. In mice where they actually injected adenoviruses with a base editor for angiopoietin like 3, they observed something like 35% edited cells in the liver, a drop in protein expression, and also promising drops in triglycerides and cholesterol in these mice. Now, these studies are actually proof of principle studies, but CRISPR is actually inching towards the clinic. 2 different approaches have been taken in clinical studies that are on the way. So undergoing studies are based on either ex vivo, harvesting hematopoietic stem cells like sickle cell disease, beta thalassemia, editing them and then infusing them back to the patient, or in vivo, more localized treatments like the eye, for example, or the liver injected with nanoparticles, and try to target very specific and rather rare diseases. very recently, only a few weeks ago, the results from one of the clinical trials that was a phase 1 clinical trial for safety and efficacy for beta thalassemia and sickle cell disease came out, and they are very promising in this setting. What they did was actually increase the expression of fetal hemoglobins to help patient, it was one patient with beta thalassemia and another with sickle cell disease. So promising findings, but still very early on. In conclusion, CRISPR technology can be a powerful tool for cardiovascular research, but to realize its full potential, we must establish strategies to ensure appropriate delivery, specificity, and repair. Finally, I would like to thank everyone that contributed to the studies that I've presented, and of course, BHF for funding our research. Thank you.
Thank you very much, Anna. We have a few minutes. Does anybody have any question for Anna? Either raise your hand or write the question in the chat.
Can I ask a question?
Yes, Alex.
Hi, Anna. It was a really nice talk. Thank you very much. It's kind of like not directly related to what you were talking about, but I was just wondering, because RNA is a susceptible macromolecule, a labile macromolecule, do you know of any research that's surrounding generating tools to limit its degradation? So RNA, like stable RNA binding proteins or something that can coat RNA and protect them from being degraded. I'm thinking about this more in the recent Pfizer vaccine, coronavirus vaccine, and how that's an RNA vaccine, and just RNAs in general, and the tools for CRISPR-Cas heavily rely on RNA.
Yeah, Alex, I'll tell you what, when it comes to CRISPR, you actually don't want things to stay around for too long. If possible, you need a very short time frame so that the cut occurs and then everything is degraded. Otherwise, think of it as two pairs of scissors that are there all the time. And even if there are some mismatches and there is not a perfect homology, still, you have so much time in your hands, you may as well cut the off target. So this is why the RNPs, the ribonucleoprotein complexes, are sort of the ideal tool, because your half-life of RNA and protein, they will only suffice for you to have the break, and then they are gone. So I know for what you are saying in terms of stability, from the RNA, microRNA field, I know there are several modifications that they do, and I'm sure that they incorporate like phosphothiourate bonds on this RNA molecule. But when it comes to gene editing, I can tell you that time is not on your side. We want it, quick.
Yeah, I mean, I can appreciate that. I was thinking more from the vial to the cell or to the organism. So it's really that idea of storage, transportation prior to use, because that's the sort of point at which it could become, unstable. Obviously, when it gets into the cell, you would want it to dissociate.
Yeah, this is a good point, and actually there are limitations. It's not only in terms of how intact it is, but also does it have the right conformation. Because the adapter molecule and your guide RNA have to have the right conformation for you to actually have a functional molecule. So a few years back, the companies couldn't even sell the entire thing as one piece. It will give the two pieces so that you then compile the complex. Now I hear that they've come with modifications that they made so that it is stable, and you can buy the whole thing as single guide RNA and not as CRISPR RNA and tracker RNA anymore. But you're right, this is one of the limitations. I think what we basically do is, just flood the system with RNPs, hoping that some of them will work, and that will be sufficient.
Mm-hmm. Thanks, Anna.
Okay. So I think that Joseph has a question. No?
Hi, Anna. Yeah, great talk. I have a quick question whether the efficiency of the cutting and things is higher in proliferating cells, because how does the guide RNA access the single-stranded DNA to make the cut? If that makes sense.
Your Cas9 is the one that will actually open up the DNA. So what is happening is your Cas9 by itself is an inert molecule, or is considered an inert molecule. So it won't even bind on your DNA without your guide. It's because you have your guide, it will bind on the DNA and start interrogating the sequence. But it's only the presence of PAM that will allow it to cut. But your Cas9 is the enzyme that actually will open up the DNA, so that you have the two strands separated, and it can cut each strand with one of the nucleus domains.
Perfect, thank you.
Any other question for Anna? No, Joseph, you have the hand raised, but I think is the old one. Yeah. So if there are no other questions, thank you very much, Anna, for your help today, for your great talk.
My pleasure. Thanks a lot.
Thank you very much.
Thank you.